Great. Hi, everybody. Thanks for coming. So, as was probably said before, feel free to share these slides and adapt them to your own liking, as long as you have proper attribution and licensing. So this is Module 6, where I'm going to be talking about cell-cell communication. Over the course of this lecture, I hope you'll take away the role and context of cell-cell communication, how we can use single-cell technologies in order to extract cellular transcriptomes in order to better identify communication, some general principles about how most of these algorithms can work, some of the more detailed inner workings of a select few communication detection algorithms, how we can maybe improve the robustness of this detection through spatial transcriptomics, and lastly, ending with the creation of one of our own, from our own lab, spatially resolved cell-cell communication tools. So how do cells communicate? There's a variety of different kind of mechanisms. It's not through cell phones. It's generally through the ligand receptor pairs. So basically, people can see the mouse. Is this? Through a sender cell will secrete a ligand protein, which will bind to a cognate receptor on a receiver cell, and this will form that communication. So there are many different types of mechanisms for this ligand receptor pair based communication. For example, you can have autocrine, where the cell secretes ligand that's picked up by its own receptors on the cell surface. There's paracrine, where the ligand can travel a short distance and bind to the receiver cell a short distance away. There's juxtacrine, where there has to be direct cell-cell contact. And endocrine, where the ligand can travel through the endocrine system a very far distance away within the body. Usually this is through hormones. And so why do we care about all this communication? Well, upon binding to a receptor on the cell surface, this will initiate a cascading event of signal transduction, where one protein will activate another protein, which will phosphorylate another protein, which will then form a complex and activate more proteins. And it can even translocate into the nucleus through, for instance, transcription factors, which can regulate gene expression. This can have major effects within the cell, altering its behavior through different pathways such as apoptosis or cellular proliferation or initiating an adaptive immune response, all these different kinds of functionalities. And so you can imagine that the dysregulation of these communications can lead to conditions such as different kinds of diseases. For example, in my lab in particular, we're interested in cancer and how the dysregulation of these signals can cause resistance to therapy. So you could imagine having a very diverse tumor with many different cell states and cell clones. Upon treatment with a drug such as chemotherapy or targeted therapy, while most of these cells may be eliminated, there might be a few that survive into minimal residual disease, maybe from cell-cell communication. And upon a drug holiday, it might grow out and cause relapse. So as a result, labs like us are very interested in cell-cell communication and how it plays a role in this kind of behavior. And we hypothesize that this is happening because what we notice is across different kind of models that we used in xenograft models and in cell lines, across colorectal cancer and T-cell acute lymphoblastic leukemia, when we look at the most differentially expressed genes between the minimal residual disease, the treated cells versus the control, we see that some of those most differentially expressed genes are involved in cell-cell communication. Furthermore, a lot of our past projects where we create different kinds of tools, whether we're looking across different kinds of cancers or we're looking at the same cancer across different individuals, or within even a single cell line or a single cancer sample, there can be a lot of this heterogeneity. A lot of the diseases that we've covered, such as acute myeloid leukemia or T-cell acute lymphoblastic leukemia, are driven by mutations within their cell surface receptors. So, this kind of leads us all to believe that there's the cell-cell communication contributing to progression and this poor therapy response.